On. We're almost done here with organic chemistry. But last time we got into a couple other sort of groups, or really the main sort of one we talked about there was amines. And again, these guys here are nitrogen containing. They are derivatives of ammonia. So they typically have a nitrogen and usually a couple hydrogens. Sometimes they'll have less hydrogens. Again, uh, kind of the more carbon groups you put on there, you do have to kind of get rid of a hydrogen to do it. And the way that we are naming amines is basically just the common name. And again, like I said, sometimes people will not draw those kind of dots in there, but they are there, even if you don't see them. The common way of naming it is very much like we did with ethers. We're just going to kind of name the groups that are attached. So this would be methyl. And again, we would just put amine out there. If we had a couple of these, this would be a methyl and another methyl. And again, you can see we lost the hydrogen there by putting an extra carbon group on it. And that would be dimethyl amine. And obviously, let's and here we would have an ethyl group and two methyl groups. And this would again be alphabetical order. So ethyl, dimethyl, and once again, just put in the amine at the end. We also could classify amines as primary, secondary, and tertiary. And again, we're looking at the nitrogen and how many carbons are directly attached to it. So in this case, it'd be a primary. Here, the nitrogen has two carbon groups attached. That's secondary. And here, the nitrogen has one, two, and three groups attached. And that would be a tertiary amine. Again, the hydrogens go down every time we put an extra carbon. They kind of start with two hydrogens because it's assumed that they already have a carbon group attached to it. As you put another carbon group, you do lose the hydrogens on it. We also saw that we could have our benzene ring with that NH2 group, and that again is aniline, that sort of historical sort of name. And we also saw in this particular sort of situation, we could have a group that's actually attached to the nitrogen and not the carbon. And remember that when that happens, we actually don't use numbers for it, but we do use N to indicate that it's attached to the nitrogen. So this would be something like N-methyl aniline. And we still could have, though, groups attached to the carbon, like here. This would then be a number situation where the nitrogens attached would be carbon number one, and that'd be carbon number two. This would be 2-chloral aniline in this case. So uh, again with this, and it can happen actually with the sort of chain ones, not the ring ones, but you can sometimes have groups attached to the nitrogen versus the carbon. And again, in terms of the naming, we do use that N instead of numbers. And as we saw last time, you could even have two groups attached to that nitrogen. And again, much like numbers, they do like an NN type situation to indicate that they are both attached there. Any questions on that there? All right, so I think where we left off is here. The very last organic group of the season here is the amides or amides, and they have a functional group that looks very much kind of like a carboxylic acid and kind of like an ester group, except it is actually bound to a nitrogen here. And Tylenol, for example, is acetaminophen. That is an amide. And here you could kind of see the amide group right there. Whoops, come back. And that is, again, the functional group of it is that connection. You could probably maybe guess how you get one of those, and you get one of those basically by carboxylic acid and an amine coming together. So that's the amine is going to provide nitrogen. Carboxylic acid there is going to provide basically the carbon oxygen double bond in this particular case. And that's what we see here. Again, we have an amine. Well, that's our amide, I'm sorry. And we have our carboxylic acid. And again, here, they're going to come together with that NH3 group there and going to basically form that. So you're going to get basically your kind of carboxylic acid and an ammonia derivative like an amine happening. And basically what's going to happen is, like most things, we're just going to take out water, basically. There's our two H's in our oxygen. And basically going to throw everything together. I'll put a carbon at the end of this guy here. And we'll end up with our amide connection here, which again is right here. So again, they're made by this reaction here. This is actually a really important sort of bond as well. And we'll talk about it a little bit in protein chapter. This is really a peptide bond. So this is a peptide bond. This is how amino acids come together and make that bond together. When two amino acids come together and bond, they make basically this amide bond here, this mid bond. And that's basically what's sometimes referred to as a peptide bond, where you talk about amino acids coming together to make that bond. Because amino acid basically has both of those groups. An amino acid basically has an ammonia type group, and it also has a carboxylic acid type group. And again, those guys come together, and when another one comes together, they basically make this type of bond. And that bond, again, is what is sometimes referred to as amidation reaction. It's a fancy way of saying we're making an amide bond here, basically. And as I just mentioned before, we're basically just going to take water out, and from the OH from our carboxylic acid and the H from our ammonia or amine, that is what's going to produce, again, the water that comes out. And basically all we're just going to do is slap both of those sides together. And once again, the connection point is going to be the functional group, that carbon oxygen double bond to the nitrogen hydrogen in this case. So that was with ammonia. You could also do it again, like I kind of demonstrated a second ago, with an actual amine, and again works the same way. We got our carboxylic acid and we got our amine, and if you just kind of back them together, hydrogen to hydrogen, that's where your water is going to basically come out, and again, just going to squish everybody that's left basically together. Absolutely nothing else changes. You got your CH3, your CH3, your CH2, your CH2, your carbon oxygen double bond, your nitrogen hydrogen, and your CH3. Absolutely, again, nothing changes other than you took water out and basically squished everybody together to make that bond. All right, so if we were going to do this one, once again, if you're going to sort of draw this, sometimes it's helpful, like we did with some other things, to maybe turn your amine around so that, again, the hydrogens are sort of facing each other. And if you did that and you just sort of redrew it, and I'll even draw the hydrogens out here, and we'll go like that. So now all we have to do basically, again, is just kind of box out the water. And once again, nothing else is going to change. Everything's going to be exactly the same. So I'm just going to copy everything the same. And in this case, I actually drew the lines there on benzene. We'll have our carbon, oxygen. Obviously, at this point, we pretty much got rid of all this. So now we're going to connect that carbon and oxygen double bond to what's left on the other side, which is our nitrogen, hydrogen, and our CH3 in this case. So a very similar type of reaction that we've seen before, where a lot of things you just take water out and kind of squish everybody together, is sort of what ends up happening in these sort of reactions. Obviously, water would come out on this side if you wanted to as well. And again, that is right here what forms our functional group there and our bond. Any questions on that one there? Now, naming amides, we're not going to do, so that's good. Still not going to do it. Even though there's 3 slides, still not going to do it. Still not feeling it. I would give you the name, but I don't feel like doing it. There we go. It's not doing it. Let's talk a little bit about amides. Urea, again, is end product of some protein metabolism removed from the blood by the kidneys. It also is, you can see here, here's our amide bond that's happening in that particular case. A lot of barbiturates are also amides. Again, you could kind of see those bonds happening there or wherever you want to kind of look as well. Aspirin, and again, this is in line structure, right? So there is a carbon here, so that's our carbon oxygen double bond, and that is our nitrogen hydrogen connection there that gets our bid there. And as we saw earlier, acetaminophen here, same thing. There's our carbon oxygen bond connected to our nitrogen hydrogen, again, also an amide. So let's just talk about a couple reactions that amides will undergo, and it's very similar to some of the reactions that we saw before. There's a hydrolysis reaction, and there's an acid hydrolysis and a base hydrolysis, and kind of similar to sort of what happens in some of these cases. We are going to, as you could probably imagine, break this guy right at the functional group, and that's going to give us our parts that are left over, basically. And in this case, in the acid hydrolysis, we will get the acid back. So this is a carboxylic acid part, and this is an amine part. And again, the way you can remember that is in the acid one, you get the acid back. So you can see you get the acid completed back, and that is basically just putting the OH onto it. So if you want to just keep track of it, take the OH out of there. That then leaves us basically a couple of hydrogens, and those couple of hydrogens find their way back onto the other piece here. And we get a couple of hydrogens, whichever ones you want to look at, those two onto there, onto that piece. And obviously we have a chloride that's left over, and it's just floating around. So in the acid hydrolysis of an amide, you will get back to completed carboxylic acid, and you will actually make an ammonium salt. So you won't get the amine back perfectly, but you'll make sort of a salt by adding those guys in. And the acid here is providing some extra hydrogens that's necessary for that to occur. Now in the base one, again, we're going to break right here. In the base one, we're using some type of strong base like sodium hydroxide or potassium hydroxide. And in the base one here, we actually get the amine back. So you can think of a carboxylic acid, obviously, as an acid. A lot of amines, as we talked about, are bases. So these are good ways to remember sort of what you get back. If you do the acid hydrolysis, you get the acid back. If you do the base hydrolysis here, you're going to get basically the base back, and that's going to be the amine part of it. And that's what we see here. When we kind of broke it apart, we had everything over here on this side. And we're going to complete our amine by taking out our hydrogen here. And that's going to end up over here when we do that. And what's left is an oxygen and a sodium. And that ends up at the carboxylic acid side. And again, we're not going to get our completed carboxylic acid back because we actually need the acid to be added to it to actually do that part. So these are two little reactions that they kind of undergo. And that's sort of a good way to sort of remember sort of what you get. Acid, you get your acid back. If you do the acid hydrolysis, base, you will get the amine back, which is essentially a base. And that's it on that, I think. Don't worry too much probably on that there. All right. We don't have to draw the structures because we're not naming them, so that's good. And I feel like I'm out of lecture on that there, so that's good. So for amides, you really just need to know the functional group and maybe how they're made. That would be good, I think, yeah. So I wouldn't worry too much about the hydrolysis reactions for those two. Okay. All right. Wraps up organic chemistry for the season. Deal. All right, we're gonna go backwards now and kind of pick up with carbohydrates, which is chapter 13. So we'll talk about some bread, some pasta, just in time for dinner. Right, there we go. Those are hands. Good, good talk. Still hands. There. So let's first talk about chiral compounds, and we'll explain why there's hands here, I guess, in a second. But chiral compounds have what are sometimes referred to as a chiral center or a chiral carbon. And basically, a chiral carbon is a carbon that has basically four different groups attached to it. So it is a carbon that basically has four different groups that are attached directly to it. And we'll see this in some carbohydrates coming up. And because of that, that is what makes it a chiral compound, because it does contain a chiral carbon. You can, by the way, have more than one chiral carbon in a particular compound. There could be multiple ones. And the deal with chiral carbons is if you stick a chiral compound up to a mirror, you will get a mirror image that is not superimposable on the original structure. So that means if you take out the mirror image, you could rotate that mirror image as much as you want in any directions. There will always be something when you go to put it on the original structure will not line up. So they will not line up perfectly. Hence the hands demonstration here. If you take this hand and put it up to a mirror, you get this hand staring at you in the mirror, right? And if you pull out this hand from the mirror, I could do whatever I want with it, rotate as much as I want. I will never be able to get them to sit perfectly on each other. I got thumbs on the correct side, but palm up, palm down, so that's not going to be right. Now, if I rotate it around the other way, again, still not going to work. So you can do whatever you want with your mirror image. You can rotate it as much as you want. There's always going to be something that's not going to fit perfectly on top of each other. Clearly, if I took my mirror image, flipped it the other way, now I'm palms to palms, but thumbs are not on the right side. So there's always something that will be sort of incorrect. So if you have a chiral compound that has basically a chiral carbon in it, it has four different groups attached. There will always be, when you put it in the mirror, its mirror image will not superimpose perfectly. Now, there is something that is known as a achiral compound, and an achiral compound is different than that. And in an achiral compound, the mirror images do superimpose on each other. So in an achiral compound, you do not have four different groups attached. You got at least one group that's the same. And because you have one group that's the same, when you pull out the mirror image, you can actually rotate it around and get it to fit perfectly on the original structure. So if we look at this guy here, these are mirror images of each other. And again, if we put this original guy in, it's going to reflect back there. This guy's in the back, so he reflects in the back, and this guy's here. If we took this mirror image out and tried to superimpose it on the original guy, you can see you could rotate as much as you want. You could get a couple of things to line up, but these guys will kind of always be in the wrong orientation because of they really won't fit perfectly on that. And again, that is a chiral carbon in the middle, as it has a hydrogen that's different than a chlorine, that's different than an iodine, and that's different than a bromine. So those are four different groups basically attached to it. As I mentioned a second ago, an achiral structure or carbon or compound is one where you basically have at least probably a couple groups that are the same. So if we take a look at this one here, this is going to be a achiral carbon. And that is because here we got obviously two groups that are the same. So it does not have four different groups attached. So when we put that into the mirror as it reflects on the other side, and this guy's in the back, so he ends up in the back. And if we take that mirror image out, you can rotate that because really we've got two groups that are the same, and it will fit perfectly on the original structure. So if you have a chiral compound, its mirror image will not superimpose. If you have an achiral compound, if you take the mirror image out, it will be able to, with some rotation, be able to fit perfectly on the original structure. Question. Here are some examples of chiral and achiral stuff. Chiral are like gloves. Chiral are golf club. Achirals are soccer ball, are glass there. Um, and our shoes are chiral as well. And so right-handed scissors. Anything with like a plane of symmetry, if you could cut it in the middle, it's basically the same on both sides, will have be basically achiral, right? And they can superimpose on each other. And that's what's happening with like the soccer ball and the glass in that case. All right, so for each of these, decide are they a chiral compound or an achiral compound? All right, let's take a look at the first one. So really here, we want to go to the carbon. Um, because we usually are looking for the chirocarbon, and we really just want to look at everything that's attached to it and see are they different. And obviously here our chlorine is different than our methyl, and both of those are different than our ethyl, and obviously all three of those are different than the hydrogen. So those are four different groups that are attached to the carbon. That would make it a chirocarbon, that guy in the middle, and that would make this a chiral compound. And that means that this mirror image would not superimpose on each other because of that. If we take a look over here, again, we look at the carbon. That chlorine is different than the methyl is different, and the chlorine is different than the hydrogen, but that looks like two of the same. So we do not have four different groups attached to that carbon. So that's going to make that carbon an achiral carbon, and that's going to make this compound achiral as well, since that's the only carbon that's in this compound as well. That means if you took that guy and put it up against a mirror, you could take the mirror image out and it will fit perfectly on the original structure. Lastly, here, if we take a look at this guy, we got one group methyl different than the chlorine. Those two are different than the hydrogen, and all three of those, again, are different than the bromine. So this would be a chiral carbon. Any questions on how to identify those there? Okay. And like I said before, in each of these cases, there's only really one carbon in this compound. And because there's only one carbon, that will basically tell you what it is, the overall compound, chiral or achiral. As I mentioned before, you can have, definitely with carbohydrates, as we'll see, multiple carbons in it. And you can have some carbons that are achiral in the compound, and you can have some that are chiral as well. And you can have multiple chiral carbons as well, and multiple achiral as well in a compound. All right, so hopefully they agree. That's good. All right. Now, Fisher projections are sometimes used to represent three dimensions. We're not going to do too much of it in here, but in case you see it, in two-dimensional representation, usually the most oxidized group is on top, and that's usually the group that has oxygen in it. We use vertical lines in place of dashes to represent things that go kind of behind the paper. And typically, if you see dashes, that's also what it represents, kind of three-dimensional, like behind the paper. We use horizontal lines and vertical wedges or wedges to indicate things that are coming forward. So sometimes you'll see drawings where they'll do like lines like that, where that group is kind of going behind the paper, and then sometimes they'll have a wedge that it's just sort of coming out of the paper and stuff like that. And that's sort of what we see here. This also is another thing that's very common that we see, and a lot of times carbons or sometimes chirocarbons are represented almost like a line structure when we're dealing with carbohydrates, where it kind of crosses. This is usually a carbon. So that's a very common sort of shorthand that's sometimes drawn for these things, just kind of lines across the lines. That so kind of like line structure where goes up and comes down. Everywhere it kind of crosses each other is a location of a carbon. And that's what we see here. This is obviously a carbon. This is our most oxidized group, which has the oxygen in it. And this would be a carbon. And then obviously this would be a carbon as well on the bottom. Now, another designation that we sometimes see with things, especially also carbohydrates, is the DNF notation. And basically what that means is D means right and L means left. And it's the location in this case of the OH group on the last chirocarbon, say in a carbohydrate. So the last chirocarbon from this sort of oxidized group up on top, the last chirocarbon down the road is basically which side you find the OH group there. So for this guy here, obviously, this, by the way, is that a chirocarbon, that one right there? That I just made red? That is not a chirocarbon. It doesn't have four things attached. It actually only has three things attached, right? It has one thing here, and then basically everything below what I just drew there is a group. This is a chirocarbon. That chirocarbon has this guy, it has everything up here, and it has everything right here. So those are the four groups that are attached to that chirocarbon. How about the carbon down here? Is that a chirocarbon? That is not a chirocarbon because if you kind of draw it out, everything that's on there, it has, everything up on top of that carbon as a group. It has obviously the OH as a group, but it also has two hydrogens there, right? Which means it's more an achiral carbon, right? Rather than a chirocarbon. And that is typically in carbohydrates, kind of the group you see at the very end. Uh, it's always that group. And I won't say 100% of the time, but pretty close to it. If you back up one carbon from there, that's usually the very last chirocarbon in a run. And that's a good place to look at it. So in this particular case, when we look at this, this is really the only chirocarbon in this compound. And the OH group here is on the left-hand side, and that is why it's called L-glycerolaldehyde. And obviously, if we take a look at sort of the one that's related to it, again, this is our only chirocarbon in it. The OH group here is on the right, and this is what is referred to as D-glyceraldehyde. It is the same compound, except that the location of that OH on the last chirocarbon is in a different sort of location. One's on the left, one's on the right, which does give it certain different properties in certain cases. Any questions on that there? All right. So for each of these, indicate whether the mirror image cannot be superimposed. It's like a double negative, I think. I don't know. Or not. You really just got to decide, are these things really chiral compounds or achiral compound? If they are chiral compounds, then their mirror image will not superimpose. If they're achiral, then it will. So we will just look at, since these are mirror images, you can think of the mirror as being pretty much right there on that. So if this was our original structure and on this side is our mirror image, if we take a look at our carbon, this group is different than that group, and those two groups are different than that, and all three of those groups are different than the other one here, the bromine. That would mean that is four different groups that are attached, and that would make this a chiral carbon. And once again, since it is a compound that has a chirocarbon, this would be a chiral molecule. And that would mean that its mirror image would not superimpose. So it would not superimpose. But not to confuse anybody, to answer the question, the answer would be yes for that because they cannot superimpose, because that was the question that was asked in this case. If we take a look down here again at the carbon, this group and this group are different, and those two are different than the hydrogen. But once again, we do run into a second hydrogen there, which means that we do not have four different groups. That would mean that this is an achiral carbon. That means that when we take this mirror image, we could take this guy out, we could rotate it, and we could actually get it to sit perfectly on the original structure, and this guy would superimpose. So, any questions on that there? Now, again, to answer the question, it was asking which ones cannot superimpose. So That is why it says yes to the first one and no to the second, because yes, it cannot superimpose, and no, it cannot superimpose on the second one. All right, let's talk a little bit about carbohydrates. Carbohydrates are a main source of energy. They are made up of carbons, hydrogens, and oxygens. We take in some carbohydrates, go through a little glycolysis, right? Use it for some energy, store some for later, in case we need to break it down through a little gluconeogenesis and break down some of that stored sugar. And in this particular case, they are sometimes called saccharides, which means sugar. All the yummy stuff right there, bread and pasta and all that good stuff, right? Now, carbohydrates are produced through photosynthesis in plants, such as glucose, which is synthesized from CO2 and water and a little bit of sunlight, and this nice little respiration cycle here, and making our glucose from our CO2 and water with a little bit of energy needed. Now, when we talk about carbohydrates, there are a couple of different types of carbohydrates we're going to talk about. There are monosaccharides, which are the simplest type of carbohydrates you could get. They're basically made up of one sugar molecule. There are disaccharides, di meaning two, so they're basically made up of two sugar molecules together. As we will talk about how you put them together, basically is you take water out and then stick the two sugars together, basically. And there are polysaccharides, which are really long groups of sugars hooked together through certain types of bonds. And we're going to talk about each of these here. When we have a monosaccharide, because there's only one sugar molecule, you can't really break it down any further. If you have a disaccharide, basically, since water is what is sort of removed to make it, if we wanted to break it back apart, we put water back in and we would break it apart into as simple sugars. And if we had a polysaccharide, which is really just a bunch of them hooked together through the removal of water, if we tossed in there a bunch of waters, we could break them up into the individual sort of sugars. So let's talk a little bit about monosaccharides first. Monosaccharides consist of usually 3 to 6 carbons typically. They have really one of two organic functional groups that we've seen. They will either have like the aldehyde group or they will have the ketone group. So a reminder, hopefully it hasn't been that long. This is the last chapter, I think. This is our aldehyde group. And again, this is our ketone group, which once again is our 3 carbon run. And that's going to be our ketone group. So these monosaccharides will really have one of these groups, and this is usually what is considered the most oxidized group. So they're usually drawn sort of the top of the monosaccharides when you draw them. And just like in organic chemistry, because the aldehyde group has that hydrogen, just like an organic, it has to be at the end. It will also be at the end here of a carbohydrate because of that hydrogen. And that carbon-oxygen double bond will kind of be carbon number 1. And just like in organic chemistry, because you need a 3-carbon run for the ketone with the double bonded oxygen in the middle, in a carbohydrate that has a ketone group, that carbon-oxygen double bond will always probably end up at carbon number 2 because you need kind of that. And just like in organic, when we number the carbons, and we do that with sort of our carbohydrates to help us draw it and stuff like that, we always start numbering from these groups. And kind of like in organic chemistry, they kind of take the priority in terms of numbering. We do want to give these sort of carbon-oxygen double bond kind of the lowest number. In the case of an aldehyde group on top of a carbohydrate, it will be carbon number 1. And usually in the case of a ketone group, it'll be carbon number 2, which would give it that. And then all the other carbons that may be on that monosaccharide, again, are sort of numbered accordingly. And like I said, this group is usually at the top. These groups are usually placed at the top. And as we saw in that other picture, usually at the bottom, you end up with that group towards the bottom of a carbohydrate. So you kind of have like the aldehyde or ketone towards the top. You usually will end up with the CH2OH at the bottom. And in between will be a variety of however many carbons, depending on the carbohydrates that you have. They'll kind of fill in the middle of it. And again, in between, you usually have, some hydroxide groups hanging out there. And usually on the other side of the hydroxide groups are hydrogens. And those are kind of the major way that a carbohydrate sort of looks. So let's talk about the one that has the aldehyde group on it. And the carbohydrate that has the aldehyde group is sometimes referred to as an aldose, as the sort of type of carbohydrate it is, because it does have that aldehyde group on it. And we could also classify the carbohydrate based on how many carbons it has. Triose would have 3 carbons. A tetrose, like tetra, which is 4, would have 4. Penta is 5, right? So pentose would have 5. Hexa is 6. So hexose would have 6 carbons. So when we look at this particular one here, we would have 1 carbon there, 2 carbons there, 3 carbons there, and 4 carbons there. So this would be an aldose tetrose, as it has 4 carbons in it, and it has the aldehyde group, right? And when we look at this one, how many chirocarbons does it have? So take a moment, look it over, try to figure out how many chirocarbons out of those 4 are chiral, if there's any. We go with none because I said there might not be any. Is that what we want? That's like the all of the above answer on the multiple choice, or the none of the above. Let's take a look and see. Well, let's start with this one here. Does this one have 4 different things attached to it? Does not. So that's gonna not be obviously a chirocarbon. So now we come down to our next one. Does this have 4 different things attached to it? It does. So the 4 different things, in case you're not sure, is this guy, this entire thing, and that entire thing. Yeah. So that is 4 different things, and that would be a chirocarbon. Now, if we go to our next one, is our next one a chirocarbon? So let's go to this guy. Let's just go that color. Is this one a chirocarbon? This one is a chirocarbon as well, because it has this group, this group, this group, and now this entire group, which is different. And all 4 of those things are different. So that is a chirocarbon as well. And lastly, if we head down to, but don't erase the whole structure, maybe. Let's see. There you go. We head down to the bottom one. Is the bottom one a chirocarbon? The bottom one is not a chirocarbon because, again, it has 2 hydrogens. Yeah. So because it has those 2 hydrogens, it cannot have 4 different things. It does actually have 4 different things attached, but 2 of those 4 things are hydrogens, and that would be more an achiral carbon. Would this be an L or a D? It would be a D because we're looking for the very last chirocarbon, which, by the way, would be this one. Usually, like I said, almost right above that guy usually. And our OH group here is on the right-hand side. So this would be in sort of the D formation in this case. Any questions on that? Yeah. In order for it to be a chirocarbon, it does have to have 4 different groups attached. Yeah. Exactly. It's got to have exactly 4. So for example, the one on top actually only has 3 things attached. And this carbon right here has this guy, and then basically that. So it only got really 3 things. So it's actually kind of neither. So it's gonna kind of, it's not a chiro or an achiro. Yeah. Correct. So when we look at those individual carbons attached to say, so attached to say the green carbon there, we would just be looking at sort of that green carbon of what's sort of attached to it, not looking at everybody's sort of individual pieces. So just attached to that green carbon would be this guy, this guy, this guy. And now pretty much everything below it would be kind of like a big group now because we're just looking at that carbon. And then that's what happens when we move to this carbon. When we move to this carbon, when we look above this guy right here, now becomes part of everybody behind it or above it. And that all becomes kind of like one group. And we look at it all the way towards the end. And that's why that ends up being a chirocarbon as well. Because so a lot of times people will look at it and go, well, it's got like the same group right there, right? As you know, for the one above it or something like that. And again, they carbon above it now becomes kind of attached to what's behind it or above it as well, and it becomes one big group at that point. Exactly, yeah. So, and that's why when we look at this carbon down here, the four groups for like this carbon down here, even though it's not chiral, is basically it's basically this OH, these two hydrogens, and then basically everything above it would be considered the group that's attached to that particular carbon. And that would obviously be different than the other things, but in this case, we have the two hydrogens and stuff like that. So every time we kind of move down, anything above it now becomes kind of the group that's above it with everything else that's above it and stuff like that. Yeah. What we're looking for in terms of D is what we're looking for. Actually, let me just erase everything that's on there. It's got a lot on there now. There we go. What we're looking for is the very last chiral carbon from the top, where this group is, where I just boxed. We're looking for the very last one down the road that is chiral. And then once we find that carbon, we're looking at which side the OH is on. And in this case, it's on the right-hand side, so that would be the D. If it was on the left, it would be L in this case. And like I said, maybe not 100% of the time, but pretty close to it. If you just go to the bottom and find this group and you just kind of go back one carbon, that's usually in most cases, again, not 100% of the time, but in a lot of cases, that's going to give you probably your last chirocarbon and down the road because that CH2OH group on the bottom is usually always on the bottom and you're looking for the really last one from the top. So in most cases, if you just kind of back up one, that's usually going to be, like I said, 90 some odd percent of the time, or even more than that, is going to be probably your last carbon. Yeah. Yeah. What's that? Up here, it's not chiral because it doesn't actually have four groups attached to it. So it's actually you can think of it more like it's kind of like the functional group, if you will, in this case. And it's not, if anything, it could be considered achiral, but most people probably wouldn't. It's because usually we're looking for kind of four different groups for it to be chiral. So it's definitely not chiral, and it's more a part of the functional group of the carbohydrate. I guess. The double bond doesn't matter. Yeah, we're actually looking for like things attached to it, if that's what you're asking. So, like I said before, typically this is what one thing that's attached to it directly to the carbon. This is attached to it, and now again, everything below it is kind of one big group that's attached to it, and that gives it only three things that are attached. So it's not necessarily. It has nothing to do with bonds. It has to do with how many sort of atoms or things or groups of atoms that are attached to the carbon directly. Other questions? Okay, so this is an aldose. It has the aldehyde group. Again, it was a tetras because it has four carbons. We also, as I mentioned and talked about there, we can have a ketone version of this, and these are sometimes referred to as the ketose. And this is our ketone group here. And here's our ketone group. It is our three carbon run with our carbon oxygen double bond. So that is the ketone group, just like we see in organic chemistry, three carbons in a row with a carbon oxygen double bond in the middle carbon. Now, you can also see that what happens in this particular case is because the ketone group needs to, or the carbonyl carbon there is the second carbon down, we actually in this case end up with kind of matching groups on top and bottom, if you will. And we do end up with that extra group up on top. And this case, this would be a ketose. And if we count up the carbons, this would be carbon number one because it is closest to that functional group, given it's a smaller number. This would be carbon three, carbon four, carbon five, and carbon six there. So this would be a hexose because it has six carbons. And now I'm going to ask how many chiral carbons are in this one? We'll start with this one up on top. Is this a chiral carbon? It is not. It's actually an achiral, right? It's actually got four groups attached. It's got two hydrogens, which are the same, which is going to make it achiral. It's got the OH group. And then it's pretty much got everything below it as a group, right? So that's the four groups that are attached in that case. So this would not be a chiral carbon. This is more our functional group. Again, doesn't really have four things there, so we're not going to really worry about it. How about this carbon? Is it chiral? It is chiral, right? Again, it has everything above it as a group. It has the OH, it has the H, and it has everything below it as a group. So that would be a chiral carbon. How about the one below it? The one below it is a chiral carbon as well. Again, it has the OH group, it has the hydrogen, it has everything above it, which is different than everything below it. So those are the four groups right there that I put in boxes for this particular chiral carbon, which is right there. Put that guy right there. So all those guys in blue boxes are the four different groups that there are for that particular chiral carbon. And again, that it's a very common thing that happens to people when they look at it, like I said, because they see kind of like the same thing repeated, like carbon OH, carbon OH, and they just kind of focus on just the next thing down rather than everything else that's attached there. And that's kind of what you want to focus on. So that is definitely a chiral carbon as well. And let's get rid of some of these boxes here, maybe some of these lines. Get rid of that. All right, so then the one below that it is a chirocarbon. Again, one group there, one group there, one group there, one big old group right about there, yeah? So this guy has these three, chiral, chiral, and chiral. And the last one on the bottom is a chiral. That is achiral, right? Just kind of like the top one here, right? Those both are achiral here. So this particular one does have three chirocarbons, and this would be L or D. This would be D again, looking at the last chirocarbon, which is this, well, this carbon. And the OH here again is on the right. Yeah. Yeah. For determining DNL, what matters is the last chirocarbon from the oxidized group up on top, the functional group basically. So the one furthest down that is a chirocarbon from that functional group, whatever side the OH is on, is what determines that D or L. Yeah. For our purposes, they'll be drawn kind of like this. Either it'll be definitely left or right if that's what you're asking. So, for example, if we had, and again, sometimes they are drawn just the carbons just sort of like that. And again, each of these are carbons where they intersect, and maybe we just have like this guy here. So for example, if we had OH over here and H, right? OH, OH, H, and H. So this would be our last chirocarbon, and the OH would be over here if it was like a L determination on that. And by the way, the one I just drew had how many chirocarbons? It does have three chirocarbons. It would be this one, and this one, right? They're all chiro, yeah. No, for our purposes, it will be you maybe just be asked like, is this a D or an L sort of version of it? Would be it. That would probably be more like it. So you don't have to, and we're not going to necessarily name too much. You do have to recognize some structures, but that would be more sort of like, is this a D or is this an L sort of carbohydrate or something like that? Other questions on that there? All right, why don't we take a little break? Come on back. All right, so taking a look here at the first one, we do see towards the top here our functional group, and again, that is the aldehyde group, which would make this an aldo or an aldose. In this case, we could also count our carbons, and we got 1, 2, 3, 4, 5, and you'll get there. The 6. So that's going to be a hexen question on that one. Taking a look at the next one, we see our functional group here. This is our 3-carbon run with the carbon-oxygen double bond there in the center. That definitely is our keto group. And that would make this a keto or ketose. And again, if we counted our carbons, we got 1 carbon, 2 carbons, 3 carbons, 4 carbons, and 5 carbons. So that's going to be 5 is penta, so that's a pentose in this case. Yeah. Questions on that there. One on the left has how many carbons? It does have 4. So this one, this one, this one, and this one. And the one on the right has, yeah, it has just 2, which is this one and this one. Those are the 2 chirocarbons, I guess. Any questions? They are both D, as we would be looking at the OH here and the OH there on the last chirocarbon. They're both on the right-hand side. Any questions on any of that? All right. So let's talk about some of the structures here of our monosaccharides. Again, as I mentioned, sometimes Fisher projections are used, and a lot of times we will kind of show sometimes chirocarbons as that intersection of sort of the lines there, like just carbons in general coming through here. And here we can see this again is a chirocarbon. Our OH group again is on the left-hand side there, which would be L. This is our last chirocarbon, where our OH group is on the right-hand side. And again, that would be D. So again, the idea as we've been talking about is wherever that sort of functional group is located, which should usually be towards the top, we are looking for the very last chirocarbon down the road. And again, the location of where that OH group is going to determine that L or that D isomer of it. Here are some examples of D and L monosaccharides. You probably should put a little star on this one. You do need to be able to sort of recognize these guys, not necessarily draw them, but if you see the picture, sort of recognize glucose, ribose, and galactose. And here again, looking furthest chirocarbon from our functional group, going to be this guy right here, OH on the right-hand side, which would be D, and that would be glucose. And furthest down the road here, chirocarbon, going to be this guy, also going to be on the right-hand side, which would be ribose, D-ribose. And here from our functional group down here, we see the OH there on the left-hand side. And again, that would be our L isomer of galactose. By the way, obviously the D galactose would look exactly the same, except the OH is just switched to the other side. Everything else stays the same. Just the D would be OH on that side, H on the other side. Yeah. Yeah, so if you see a picture of these structures, be able to recognize what you will not be asked to draw it yourself, but a picture may show up somewhere like on a final or something like that, maybe, and be able to recognize what it is. So. All right, so for each of these, take a moment. All right, so taking a look here, just want to identify our chirocarbons. And by the way, for the first one, that is a chirocarbon, that is a chirocarbon, and that is a chirocarbon. So this guy would have 3 chirocarbons in it. And to identify L or D, we're looking for the last one down the road from our functional group, and that would be this carbon. And that would be an OH that's on the left-hand side there. So this should be L in this case. Same thing on the next one. This would be a chirocarbon. This would be a chirocarbon, and those are the two chirocarbons that are in here. And once again, we're looking for the one furthest down. And as you can see, as I mentioned earlier, they do tend to be kind of like right above that guy in a lot of cases. And here, left-hand side as well for our OH. So this would be an L. And lastly here, we're looking for our chirocarbons. So one there, and one there. And once again, this is going to be the last one. And that's going to be on the right-hand side there. So that's going to be an R. Yeah, I think it's an R. But I don't like R. No. Any questions on any of those there? By the way, the first two here, A and B, are aldoses, right? They have the aldehyde group on it. And the last one is a ketose or a keto one because, again, that is our three carbons in a row with our double bonded oxygen in the middle. So it has that ketone group there. Any questions on that? Yeah. Say again. That's just the name for that particular structure. So it's something you guys can kind of memorize. You're not going to be necessarily asked to name, well, put it that way. Not going to be asked to necessarily draw them or anything like that. But you do have to recognize some of the structures, like were on the last slide. They're giving, and those are just kind of the names given to those structures. Sorry. It's not like organic where you kind of follow a certain name. All right. Indeed. We'll talk a little bit about some of these a little bit, excuse me, individually here. So glucose, as I mentioned before, is a structure that you should be able to recognize if it's provided to you. So if you want to start this one with a little bigger picture, you can as well. D-glucose is found in fruits, corn syrup, and honey. It is, as we see there, an aldose. It is a hexose as well. It's got 6 carbons. It is, again, known as blood sugar. Is the monosaccharide in polymers of starch, cellulose, and glycogen, as we will see. Glycogen is stored energy, right? So you don't use it in glycolysis, it stores it, and then you go break it back down through gluconeogenesis to free it up. But it is basically in polymers of sugars or polysaccharides. They really are just repeatable units of glucose together, basically just glucose together. Obviously, blood glucose is measured right, so normal 70 to 90, and you could have one or two things, right? If you have different sort of levels of glucose, you could be hyperglycemic, right? And hyper means higher than, right? Normal blood sugar. And hypoglycemic, right? Hypo means less than, so it could have less than. And they usually measure that right when you take a blood test. So you shouldn't eat the cookie right before you do that, probably, right? And go, whoops. Or maybe that's what you say when you get your results back. I had a cookie right before I did it, right? That's probably the issue. All right, fructose, again, another structure that you should be able to recognize. And again, it is a ketose and also a hexose. It is the sort of sugar that's found in juices, fruit juices, and honey. In the body, it will actually convert into glucose. So again, may want to start those last two slides. Again, need to be able to recognize those structures. Will not be asked to draw it, but again, maybe have a picture of it. Need to be able to maybe recognize it too. Galactose, another one that you should be able to hopefully recognize. It is very similar to glucose, except that the location of the OH there on carbon number 4. And obviously, again, carbon number 4 counting from the top, that would be carbon 1, 2, and 4. Now it's 5, 6, coming down there. It is obtained from lactose, which is a disaccharide. It's not found free in nature. And lactose is a disaccharide, which is the two monosaccharides together. All right, so you're not going to be asked to draw it, so we're not going to worry about drawing it, actually. So let's talk about, though, the cyclic structure of monosaccharides and how we get there. And a lot of times, monosaccharides are shown in their cyclic form, which kind of like in organic chemistry, what essentially happens is the straight chain couple of carbons kind of come in and they kind of close up on each other, and they make more of a cyclic structure, ring type structure, rather than obviously more of a straight chain type of structure. Now, when we deal with cyclic structures of carbohydrate, there's really two types of rings that we end up with, and they're very similar to our sort of organic rings, if you will. We have one that kind of looks like cyclohexane, which is a sort of six carbon ring, but it really isn't because instead of the six carbon, there's oxygen kind of in it. So it's actually a five carbon ring because it has the oxygen inserted in. Just like an organic, right? Each of these little circles I'm making is a carbon, right? So one, two, three, four, and five carbons there. Again, it's kind of like the cyclohexane ring with an inserted in. That oxygen, as we'll talk about, pretty much gets inserted in when the ring closes. So one of the oxygens kind of gets in the middle and it kind of creates that oxygen in the ring. The other one is very much like a cyclopentane ring, which is normally a five carbon ring. But just like the other one here, it also ends up with a oxygen that gets kind of inserted inside. So it ends up being a four carbon ring. And again, one, two, three, and four carbons there. So with these monosaccharides, they kind of open up and then close the ring, and they kind of reopen and close the ring. And they kind of do that a lot, and they can end up in sort of like structure. In a lot of cases, these are very prevalent when we have five or six carbon atoms. They do form typically when carbon number five, especially in a six carbon situation, carbon number five and the functional group come together to close the ring. If it's not carbon number five, it's usually the last chirocarbon kind of comes in, and that's where it sort of happens in that particular case. So when this happens, it does make a cyclic hemiacetal. And if you remember, hemiacetals are those things that we talked about when we are talking about aldehydes and ketones. And remember that essentially a hemiacetal has an OH group, it has a hydrogen, and it has an oxygen carbon type group attached to it, and obviously some other carbon group on that side. And that's your hemiacetal. And again, it does sort of form when it comes together and makes this sort of ring type structure. And it will essentially be in this area where you see that sort of hemiacetal come together. So let's talk about how we actually go from kind of the straight chain into a ring type structure. So let's just draw a random made up sugar. So we'll go with this guy here, and I'll go this way. Go. And then we'll do this. And we'll do this. And we'll do. Okay. So we have this made up carbohydrate here, and we want to kind of turn it into obviously a cyclic type structure. Once again, as we've been talking about, everywhere those lines cross are carbons. So everywhere these lines cross, there are carbons. And if I were to, I'll throw my hydrogen over here. If I were to number or count how many carbons I have, how many carbons do I have in this case? I got six carbons in this case. Again, this would be carbon number one, right? Carbon number two, carbon number three, carbon number four, carbon number five, and all the way at the bottom, carbon number six. All right, so one way that you can turn this into a basic ring type structure is the first thing you could do is just take the picture that you got where it's trying to draw it straight up, And you could imagine yourself just like tipping this thing over, just kind of like throwing it on its side, basically. You're just going to drop it to the right. So you're just going to drop this whole structure to the right. And when you do that, if you redraw it, you'll look like something like this. And this again would be carbon number one. Again, if I drop it just down to the right, it's going to end up that way. This would be my carbon number two, right? This would be my carbon number three. This would be my carbon number four. That would be my carbon number five, and that would be my carbon number six, right? So when I take this original structure and I drop it down to the right, everybody on the right-hand side will actually end up down, right? And everybody on the left-hand side will end up up. So if I take that structure and I just go, everybody that's sitting on the right-hand side will go down, and everybody else will basically go up. So when I go to carbon number two, I have a hydrogen that should go down. I have a hydrogen that should go down on number three. I have an OH there on four and an OH on five. And obviously on the other side, those guys, as you drop down, they're going to end up pointing upwards. So anything on this side, you drop it, it's going to point up. Down effect for three, go here, and questions. So basically, I just took my guy and just dropped him on the side. Now I'm ready to do my closing of the ring. And in this sort of six carbon situation, usually, like I said, it is usually the last chirocarbon, but in this case, it's usually always number five on a six sort of run. It's going to be number five. And number five carbon there is going to find, not surprisingly, maybe the functional group there, the carbon oxygen double bond. So it's going to go and hit that carbon oxygen double bond. Now, this is a really important thing because literally at this point, you need to choose one of two rings, either like the five carbon ring like same, or carbon ring that looks like cyclopentane. How can you decide which one you need? All you have to do is when you draw a little line like I just did, is now count up how many carbons are in that. So obviously this carbon's involved, so that's one. This guy's stuck in the middle there, that's two, that's three, that's four, and that is five carbons. Which tells me I should lay up the one that looks like cyclohexane, but with the circle O in the middle there, the oxygen. So because there's five carbons involved in sort of this ring type structure, the ring that we're going to get is the one that looks like cyclohexane. It is going to be the five carbon ring. Again, obviously, if I did this and I counted up all the carbons in there and I only end up with four, I would draw the other ring in this case. Any questions on that there? Yeah. Uh, I don't know. I don't think it is. I just kind of randomly drew something to be honest with you. So, yeah, I just kind of put stuff up there. I'm not sure what it is. Yeah. Yeah. Why did I not count the last? Are you talking carbon number six? I didn't count carbon number six because it's not part of the carbons that's going to make up the ring. So the ring is going to be formed by carbon number five and carbon number one coming together. And if you do that, right, if you go carbon number five and carbon number one and bring them together, that leaves carbon number six out in the cold, right? It's not part of that. So it is not part of the ring being formed, the ring formation. So it is not counted as part of the ring because what we're counting is how many carbons do we need in the actual ring that's going to be formed. And because it goes five and one and they come together, that leaves pretty much this guy out of the ring, right? Because it's not part of it. If it was part of it, we would have started with that guy and drew it over there, and then he would have been part of it, but it's not part of the ring. So only the carbons that are starting at carbon number five and between carbon number five and one in this example, those are the only carbons that are going to end up basically on the carbon. It gets trapped in there basically, right? So like if you had, I don't have a rubber band, but like you had something that was flexible, right? And you like pinched like a rubber band, you had a little hanging off right on each side, and you put your two fingers together, you're going to have two things flapping on the other side, right? That are not part technically of that little ring circle that you would have made, and they get excluded from it. So they're attached to it, but they're not part of the main part of the ring, if that makes sense. Everybody is like between the two fingers you would pinch on that rubber band. If you kind of pulled it together, everything in between those two fingers, right, are going to be what's going to end up in that ring. And that's kind of the idea here. When those two kind of carbons come together and kind of pinch together, anything that's between those two carbons will end up on the ring. Anything that's outside of those two carbons will be out of the ring, but attached to it, but not part of the ring. Yeah, there are questions. All right. So let's talk about the poor lonely ion that we did not put in the ring, which is number 6. We always number the same way with these, which is always the functional group, the carbon oxygen double bond. What is that? It is because it's the kind of functional group of these, if you want to think about it that way. And we want to give that carbon oxygen double bond the smallest number, which in the case of carbohydrates will be either 1, like it is here, or it'll be probably 2 if it's a ketone group on top. Yeah. Say one more time. Uh, we'll talk about we'll have to talk about that. Figure it out if you had to go back to going this way. Then we'll talk about how to break it back apart. All right, so the carbon that is not part of the ring will always end up. So carbon number 6, which gets excluded here, is not part of the ring, will always end up to the left of the oxygen, and it will always end up above the ring. And this is always going to be carbon number 6 in this situation. So wherever your oxygen is in your ring, to the left of it will always be where that kind of carbon that gets left out will always end up, and usually it's number 6 in the case of 6 carbons, and it will always be to the top left. That is also important because that's going to give us our numbering of everybody so we know which carbon is which. So if that's number 6 and we know we only have 6 total carbons, right? We're going to now drop down to the ring, and this would be carbon number 5. And again, we would go counterclockwise here. This would be carbon number 4, carbon number 3, carbon number 2, and carbon number 1. And as we were just talking about, what made this ring sort of come together was carbon number 5 and carbon number 1. And that is basically this connection here. That is where the ring is closing up. And by the way, one of those O's from the oxygen that's on those guys gets incorporated into the ring. So that's why there's a carbon there. The oxygen kind of gets in the way as the ring up there on that structure. All right. So now that I have that, what do I do with the rest of the stuff? Well, we the page and go to the next problem. Now, we continue on. And here's the deal. Because 5 in this case is the carbon that kind of closes up the ring, and all of its sort of stuff is actually incorporated in the green section I just highlighted. All the stuff that's on carbon number 5 gets kind of incorporated in the green section. So we skip number 5 and we go to number 4. And what we need to draw for number 4 is where the OH is located. So the OH on number 4, is it going up or down? It's going down. So you go to number 4, you put a line, and you put an OH pointing down. When I go to carbon number 3, my OH is pointing up or down. So you go to carbon number 3, you put a line, and you put the OH pointing upwards. Probably going to guess, but at carbon number 2, my OH group is going up. So I go to carbon number 2, and I put a OH there. That is how you figure out where the OH goes. By the way, on the other side of that line, which is sometimes not drawn in these, so it doesn't get so crowded, on the other side of that line is the hydrogen still. But very often they do not draw it in these structures, because you can see if you put the hydrogens on the other side of the lines, it can get really crowded. So sometimes those hydrogens are left off. But again, on the other side of these lines is the hydrogen. Yeah. Now, carbon number 1, I go to carbon number 1. That's a problem for me because, well, that's what kind of formed the ring, right? So I can't really look at it and go, is it up or is it down? Because there is one more OH that we have to put on there. And the way that we determine whether which way we put that last OH on is what anomer is referred to that they want you to draw. And it will either be an alpha or a beta. And alpha means down, beta will mean up in this case. So if I were to draw the alpha, and it's sometimes referred to as an anomer, I would put my last OH pointing down. If I were to draw the beta, it would look exactly the same, except that my last OH would be pointing upwards. So this would be OH that way. We had an OH that way. We had an OH that way. And if it was the beta, it would go up. They will ask you in the problem or tell you in the problem, draw the alpha or draw the beta. And I will tell you basically where you put that very last kind of OH group here. Animer, yeah. All right, well, I did one. You should do some. Sure you know how. I'm gonna make up something. I don't know what it is. It's just randomly drawing stuff. All right, for this one, why don't you do the alpha and draw it in cyclic form? Then why don't you also do this one? Let's do and why don't you use the beta on this one? All right, so give both of those a try, see what you come up with. Here, again, we're gonna start with the left here, and we can count our carbons. Again, we're always gonna start at the functional group as number 1, number 2, number 3, number 4, number 5, and number 6. Now, we talked about kind of dropping it on its side and redrawing it. You can, you don't necessarily have to, because again, if I have a structure like we have here, and I drop it on its side, everybody on the right-hand side will end up, yeah, hand side. If you drop it, will end up down. Everybody on the left-hand side, right? Drop it, will end up up. If you don't want to redraw it, you can just remember that left ends up and right ends up down. It's waste of time and that way. So that's what I'm going to do here. I know always, though, it is going to be, in the case of 6 carbons, the 5th carbon, or if it's less than 6 carbons, the last chirocarbon. It is always going to find the carbon-oxygen double bond. So it's always going to find that one. Now that I did that, again, I kind of have how many carbons I need in my ring here. And again, that is 1 carbon, 2 carbons, 3 carbons, 4 carbons, and 5 carbons. So that's going to tell me I basically need to draw that cyclohexane ring with the oxygen in it, which is our 5 carbon ring. So I do want to start with this ring here. And again, that means that this guy right here is not part of the ring, right, formation. So we know that that's carbon number 6. It always ends up to the left of the oxygen on the ring. So that's going to end up always over here to the left. That also is going to give us our point of numbering everybody on the ring. So that would be carbon 6. This would be carbon 5, 4, 3, 2, and 1. Any questions on that? All right, now we're just going to place everybody where they go. Once again, we're going to always skip the carbon that kind of forms the ring. So in this case, this would be carbon 5. We're going to go carbon number 4. And here I see my OH group is on the left, which means when I drop it on its side, it will be pointing upwards. So we are going to put my carbon number 4 OH upward. You go to carbon number 3, we see it's on the right-hand side. And again, on the right-hand side, when you drop it, it's going to end up down. So we are going to put our OH pointing down. Carbon number 2 there, OH group is on the left. So again, when we drop it, it's going to end up up. So we're going to end up with this. And lastly, here we are drawing the alpha, which means the last OH group should go down. Any questions on that? All right, then let's kind of do the same thing here. We got carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, and carbon number 6. Once again, given that carbon-oxygen double bond, the smaller number. In this case, go from carbon number 5 to our carbon-oxygen double bond. Yeah, always to the carbon-oxygen double bond. Now when I look at my ring, how many carbons I got on this one? 1, 2, 3, and 4 carbons only are part of it. I also end up with not just one group that's hanging off. I end up with 2 groups that are hanging off of the ring, not part of the actual ring itself. So because that's the 4 carbon that are involved, that's going to be our cyclopentane ring that has the oxygen. So we would want to start with this as our ring, right? That's going to give us our 4 carbons that we need. Now, just like we did over here, everybody on the right will end up down. Everybody on the left will end up upwards there. And just like we did on the other one, carbon number 6 will always end up to the left of the oxygen. That is where we're going to put it, just like we've done before. So we're going to go just like normal to the left of the oxygen, and we're going to put our carbon number 6 in this case. That again gives us our numbering. We come down to the ring. This would be carbon number 5, carbon number 4, carbon number 3, carbon number 2. Carbon number 1 is off the ring, which is exactly what we see here, right? It is not part of the ring. It's actually off the ring, right? So it will be actually off the ring, much like carbon number 6 is. But we'll get to that in just a second. We're just going to build it like we did before. We're going to skip carbon number 5 because, again, that's where the ring kind of comes together. We're going to go to carbon number 4. And we do see the OH group there off the right, which again means when you drop it on its side, it's going to end up there downward. So we'll go down on that. Carbon number 3 also on the right, which again also means goes down. We'll end up there. Last OH on the ring, it's determined by beta, which means it should go up. What should be on the other side of that? What's the only thing we didn't put on there yet? We say carbon number 1 we haven't put on there yet. So that's what ends up on the other side off of the ring. Yeah. Just like it is in our picture, right? Not part of the ring, it's hanging off of it. And that's where you stick it right there. And that would be carbon number 1 right there. By the way, if we do the alpha, the only thing that would be different is OH would be on the bottom and that CH2 group would be up on top, right? So when you have this sort of 4 carbon ring, because you'll end up with actually 2 groups off, that last group that's off, which is probably carbon number 1, will be determined by alpha or beta as to whether or not that will end up or down. And it'll always be the other. Questions? Yes. We would not. The double bond actually breaks as part of when the ring comes together, where it's in this case carbon number 2 and carbon number 5 come together. The double bond actually breaks. And what ends up happening is the oxygen And one of the oxygens end up here, and the other oxygen that was on, like, say, carbon number 5 ends up there. And that's where those guys kind of end up. And that's why we don't need it because it's no longer there. It actually breaks. There's a lot of stuff that happens in between that, but the short answer is it breaks, and now it's kind of like a single bond oxygen, and that's where those two oxygens sort of end up. That's on the two carbons that come together, and that's why we kind of avoid it when we kind of go around and doing it because all the OHs and those kind of that kind of either incorporated in the ring are ends up as that OH that will either be alpha or beta at the end. Other questions? This sort of illustrate that. I think the slide's here. We can maybe see that. Just find that real quick. Right here. So here's sort of what happens. So we have that OH group, for example, carbon number 5, and then here's our carbon-oxygen double bond. So what ends up happening, as you can see, the H ends up actually over here with the carbon-oxygen double bond, and that double bond breaks, and you end up with an OH. And then this oxygen from the OH group is the one that gets incorporated into the ring. So all those guys kind of move around a little bit as they're closing, and the double bond breaks, and there's some shifting that occurs in it. Other questions? You never draw it again? Correct. So a lot of times on this particular one, they actually did draw the H's on the other side, if that's what you're asking. But a lot of times it's left off because when you draw the H's, as you see in this picture, it gets a little crowded. So a lot of times people will leave the H's off. It's not wrong to draw them. But if you don't see the H's on there, it's implied that those are hydrogens. Yeah. The double bond oxygen is gone once it closes. No, there's no double bond. The double bonded oxygen actually ends up as that OH on the alpha or beta at the very end.